Okay, guys, so we're going to look at one signal transduction pathway here for right now, and that's all I'm going to videotape on. But we're going to look at actually a ton of signal transduction pathways. And we're going to try to identify like what type of receptors are involved, what type of cascade are we seeing, what type of response is going to happen. And we're going to look at all kinds of examples. So what am I saying? Well, in our outline, we did all the intro of like the basic pathway. What is a receptor like? What is a transduction like? What is the response? But there are hundreds of thousands of these signal pathways. So we can't possibly learn all of them, but we're going to learn a few of them, or you should be able to be presented with any pathway and be able to figure out, well, what kind of receptor are they using? What kind of transduction is going on? What kind of response is going on? Okay. And I want you to think of reception as there's 4 possibilities of that. With transduction, there's 2. You're either going to see protein kinases and phosphorylases, 2 of those things, right? Or that, or you're going to see secondary messengers. Okay, you might see both. Okay, but responses, there's only 2 responses. It's either going to be cytoplasmic or nuclear. And this kind of should be in its own like thing, kind of, but didn't have anywhere else to put it. So now let's look at the other side of this outline. What are we going to see this in? Well, we're going to look at plant hormones. I know it sounds like, that sounds really exciting, Mrs. Horner, but it really is kind of neat. And it's something that you don't even realize that you might see happening actually at your own house, especially the new growth in ethylene. So that's why I really like to talk about those. Tropisms, phototropism, gravitotropism, thigmotropism, which is touch. I think you can figure out photo is light, gravito is gravity, thigmo is touch, hydro, but there's all kinds of tropisms out there. And you have that one worksheet that talks about some of the tropisms out there. Then we'll get into the human hormones. That's always interesting. And it's amazing how many hormones we just, have going through us besides, the usual ones that we think of like testosterone and estrogen. Then we'll talk about the nervous system. We'll actually talk about resting and action potentials. We'll be looking at definitely synaptic type of signaling there, okay? And the communication at those synapses, how even the nerve travels down a nerve and how it like jumps from one nerve to the next. Then finally, human immune system. I love talking about that. It's nothing but a bunch of signaling. It's got to communicate for that system to work. And we're going to talk a little bit about the COVID vaccines. How are they working to get our immune system to fight off this pandemic virus that we have? And finally, apoptosis, just simply cell death. You'll pretty much look at that one worksheet that you have on that. We might discuss that in class a little bit, but that's it. All right, so those are all the different types that we're going to do. But I just want to mention one in particular that's on the back of this packet of pictures. Okay, and that's the signal transduction pathway for epinephrine, sometimes called adrenaline, right? Epinephrine is our ligand. It is a chemical substance and it is a protein. So that's why we call it a ligand. When that epinephrine binds to a G protein, it's binding to a membrane protein. So if I gave you this pathway, I might ask you what kind of reception is going on here. You would say it's a ligand binding to a G protein. I would say, is that located in the membrane or is it located intracellularly? Well, it's located in the membrane. Well, why didn't it go within the cell? Okay, why could things go within the cell? Only if they're small and oily, okay? Then the transduction. Here is that cascade or that domino effect or that whisper down the lane. We know that the G protein is involved. It has to be inactive to active. Okay, we know how that works. Adenylyl cyclase. I did spell it right in that one video, by the way. Adenylyl cyclase has to go from inactive to active. That's when that GTP runs into it and phosphorylates it. Then we have ATPs becoming cyclic AMP. Okay, that's one of those secondary molecules. So a question I might ask you is, does it use a secondary molecule here? Yes, it does. It uses the cyclic AMP. Any other secondary molecules? I don't see any. Okay, then we see a lot of these protein kinases. Okay, phosphorylation. Okay, now these are getting like inactive, active, and they're not naming them anything in particular. They keep calling them protein kinases because actually this is one of the first signal transduction pathways, and they called all these active molecules protein kinase, protein kinase. But now we have 1,000,001 protein kinases out there, so they're starting to rename them for like the different molecules that they are. Like you can see, they call this protein kinase A, okay, and then there's maybe another one. This is phosphorylase kinase, and it's still another molecule, but it's the protein kinases are the enzymes that are activating it. Remember, protein kinases activate the molecules, and phosphatases deactivate. They don't even mention the phosphatases deactivating. But eventually, when you get down to the end, so again, a question here might be, I might ask you, are protein kinases being used? Yes, they are. Are phosphatases being used? Yes, they are. Even though they're not listed here, you can't have a kinase without a phosphatase. In other words, you can't attach something with eventually detaching something. Are secondary molecules used? Yes, cyclic AMP. Okay, and finally, the response. Glycogen, remember, is a polymer. Do I dare get my beads out and show you polymer, monomer? And it's broken down into glucose, and that glucose then can diffuse out of your cells and give you that sugar rush from the epinephrine or from the adrenaline. Okay, and so that's it for this signal transduction pathway, but we'll be looking lots more, but no more videos on those. Okay, see you guys.